RBC indicate that the patient negative and no had infection. And when there is no rupture to the RBC, indicate that the patient is infected and develop antibodies against the antigens. Now we reach to the end of the humoral immune response. Now we have a new subject, which is cellular immunoassay. To do the cellular immunity and to detect the cellular status of the immune system, first we must separate the lymphocyte. And here we use the media, which is called lymphocyte separation media, or called the Ficoll Isopaque media. And it had specific gravity 1.00078. And we add the blood from the patients or any individual on this media, and then we did centrifugation, so we'll get 4 layers: plasma and platelets, mononuclear cells and lymphocytes, and the 3rd layer lymphocyte separation media at the bottom, RBCs and granulocytes. So here, this is the platelets with the 4th layer. Here is the plasma only, no platelets, mononuclear cells, lymphocyte separation media, RBC, granulocytes, and the platelets. And here the Ficoll at the bottom of the test tube, and add to it diluted defibrinated blood, and then centrifugation, and then we'll separate into 4 layers: the RBC, granulocytes, platelets at the bottom, and then the Ficoll, and then the lymphocyte, and then diluted plasma. We will take the lymphocyte because cellular immune response, in order to assess its function, we need lymphocyte. The lymphocyte, first enumeration of T-lymphocyte, and this can be achieved using the sheep RBCs, because T-lymphocyte had a receptor CD4 receptor that bind to the sheep RBC, lead to formation rosette. And we calculate the percentage of rosette cells using hemocytometer jumper, hemocytometer counter, and then detect how many from 100 cells had or got rosetting, and get the percentage. And this is the rosetting T cells. Second function, the enumeration of the B lymphocyte. As you know, the B lymphocyte had on its cell surface antibodies, and then we add to it the conjugate, and this is the direct immunofluorescence, and then examine under the fluorescent microscope, and we see the cell wall of the B lymphocyte will be apple green and fluoresce. This indicates B cell, while if it is not B cell, it's remain red in its color. And also, we calculate 100% cells and how many of them had fluorescence cell wall that get percentage. The 3rd test is enumeration of T lymphocyte subset CD4 and CD8 using indirect immunofluorescence test, and this is the T cytotoxic, and as you know, on its cell surface presents a marker CD8, and we add to it monoclonal antibodies against the CD8, and then we add the conjugate, which is consists from the antibodies coupled with the fluorescent material, and also will give us the fluoresce apple green fluorescent on the cell surface of the T lymphocyte and examine how many cells that had fluorescent cell wall and calculate the percentage. The last function is using lymphocyte in tissue typing, whether serological method or molecular method, as mentioned in the lecture, sequence-specific primer. And in serological method, which is Terasaki method or HLA typing, here we have the lymphocyte, and on its cell surface, as you know, has the HLA molecules. Here, for example, the HLA B8. And add to it monoclonal antibodies against this B8, and this is the antigen antibody immune complex. Then we add the complement that lead to fixation of this reaction and lead to activation. The classical pathway of the complement lead to membrane attack complex MAC and lead to lysis and rupture of the T or any lymphocyte. And then we add the stain, which is a three-panel blue. When there is rupture of the cell wall, this stain will enter the cell and will have blue color, while live one did not have any color because it is live and there is no destruction of the cell wall. When there is a destruction, so this means that the patients had B8. And another method, the molecular method, and we extract the DNA using a sequence-specific primer, extract the DNA from the lymphocyte, and the DNA, which is the double-stranded DNA, and then we add to it nucleotide, tag polymerase, and the primer of the specific HLA allele, and put them in the PCR, and the PCR change the temperature starting from 95 centigrade, lead to denaturation of the double-stranded DNA into single-stranded DNA, and then the machine will decrease the temperature to 65 centigrade, and this lead to annealing of the primer if it is compatible with the DNA, annealing with the primer. And then the machine PCR increase the temperature to 72 centigrade, and here lead to extension of the DNA by the nucleotide. And here we end with 2 copies, and this process will be repeated 30 times. This PCR machine repeated this 30 times. So we start with single copy of double-stranded DNA and end with multiple copies of the double-stranded DNA. And then we'll take to it gel electrophoresis, and here we find the migration of the bond that had proliferation, while it is not compatible with the allele, so there is no migration of the DNA bond. And we have a map of each allele that's put in this line, and we will get the HLA typing of the patients or of the individual. And thank you.